Thanks very much, John. So, climate change continues. Hopefully a bit of adaptation thrown in as we go along. I've been in the climate change adaptation game for a long time, and I'm going to talk about various organisms. We work on a range of different species, or we have in the last 30 or 5 years or so, and I want to try and finish up by talking a bit about the applied sense, because of course there's an urgency, as John mentioned, in terms of climate change. There's not much point sitting around doing nice genomics and metabolomics and what have you. I love that stuff, don't get me wrong. But of course, you know, we are fighting a game here we need to do something about. It's a bit like COVID in some ways. So I'm going to give you a few messages, and I'll just slip through those fairly quickly, but I'll hopefully give you some data to actually back that up. So the first one I really want to emphasize in the climate change adaptation is that we have to think about it as two limits. So we think about a hard limit, we think about a soft limit, and Carol sort of touched upon this a little bit as well. So we have situations where sometimes we have animals and plants that can adapt to climate change, and you know we can see evidence of them doing that, you know, as in the coral fish, of course. But in other cases, we just don't see it. And of course, from a biodiversity point of view, it's actually the ones that are a hard limit that are critically important. And so the question is not just describing climate change adaptation that occurs, but also trying to understand, and I think this is the question we don't really focus on very much, we're trying to understand when we hit a hard limit. So that's my system that I'll talk about a bit later. So my study system, or at least my original study system, although we've deviated a lot from that, was in Drosophila. And of course, Drosophila is one of our favourite lab organisms, but Drosophila is also quite diverse. And we have, you know, in Australia, for instance, along the east coast, we have species that occur all the way from the top to the bottom. So it's typically a cosmopolitan species, like a rubbish species like Melanogaster. And we also have native species that are actually quite restricted. So we actually do have threatened Drosophila species that might only live in a rainforest environment. So, you know, there's a huge diversity of Drosophila species. Some are threatened, some are not. Some have restricted distributions, and some have very, very narrow distributions. You know, we have species that occur in just a few rainforest patches in North Queensland, for instance. So we have this wonderful laboratory, if you like, and of course you have a fantastic climate gradient along the way. So we have a gradient going up and down in terms of temperature, mean temperature, extremes of temperature, and they don't often match necessarily, and you've also got some relative gradients going inland. So in the old days, what we used to do, of course, is to say, well, you know, can things adapt to climate? So you pick a favourite traits, and the traits in this case might be desiccation or temperature tolerance or whatever. You try and select, and you show a response, and you say, hey, things can adapt. And, you know, here's just an example of an ancient study that we did back in 1989, before quite a lot of people in this room were probably born, but just showing my age, of course. And in this case, you know, you basically have a situation where you've got some selected lines, some control lines, and you start selecting. And by the time you get a few generations, 9 generations, you've actually got a big divergence, as you can see, between those selected and control lines. So you say, hey, you know, there's genetic variation for adapting to, in this case, desiccation resistance in this particular situation. And people would then say, well, you know, this is pretty good. We can adapt to climate, no problem. Of course, this is a garbage species. You know, the rest will behave. But at least in this case, we have some adaptive potential. And, you know, that's really where we started coming from in terms of then trying to understand the sort of climate change adaptation. So we also showed us decreased water loss and what have you going on. The other thing we noticed about this early stuff is that we often got a trade-off. And, you know, I was going to ask Timothy about trade-offs and TEs, but I think, you know, trade-offs are critically important when it comes to climate change adaptation, because maybe you can adapt, but maybe something falters along the way. And in this case, you actually get a decrease in fecundity as you become more resistant to desiccation. Now, you know, that's all very well, but of course, sometimes they don't do it. And, you know, here's a couple of examples. And the one on the left is, again, an ancient study that I was involved with many, many years ago. And in this case, this is actually Drosophila melanogaster, and you're trying to push it to an extreme. So, you know, so Carol talked a little bit about temperature adaptation, but that's sort of a mean temperature. This is when you look at the extreme temperature resistance in these cases. And even melanogaster can show a nice response to selection, but then it hits a plateau. It sort of plateaus out quite early on after a few generations. So you can push it. You can push it to become about 1.5 to 2 degrees more tolerance of an extreme, a period of extreme, but then it falters out. And of course, you know, as John mentioned this morning, we are looking at extremes that are actually higher than that already. So, you know, that's the situation we face, and that's why there's urgency around us. We've also done this sort of work in collaboration with a couple of other groups. So here's another example, and this is a species. This is an alpine species, Drosophila, called Nigra sparsa. It occurs in Austria, and you can select it as long as you like for increased temperature resistance. This is high thermal resistance, and you don't get a response selection at all. And this is something that we found in a couple of other species as well. So it may be the case that, you know, some of these species are actually limited, and it doesn't matter when you push them. You know, there's no epigenetic response. There's no fixed genetic response. That's it. They just hit this boundary. So we've also shown, of course, over the years that sometimes you do get conditions where you do get changes that happen quite quickly, but then they disappear. And this is a little bit like what Carol was talking about, salinity changing. We've also got situations where we get seasonal changes happening in Drosophila. And, you know, in winter, of course, it gets colder, not too cold, but still gets a bit colder in Australia, East Coast. And, you know, we have species, native species, that become more resistant to cold after winter, after this cold period. So that's an example of Drosophila serrata. And, you know, this is work that was collected, this is data that was collected back in the 90s, but we've actually repeated this experiment a number of different years. And what happens after winter is you actually get a decrease in mortality under cold stress at that Forster population, which is the lowest point at which you get the species after winter. If you then go to the next year, you know, before the next winter hits, this is pre-winter, you find that actually that mortality is exactly the same. So what this is showing is that you're getting temporally based selection. These are all genetic-based differences. It's common garden stuff. So in this particular case, you're increasing the resistance, and then, of course, that resistance increase disappears over time. So what we think here is that this is an example of a hard limit, but this is an example of what we call a soft limit. And in this particular case, if you actually measure the gene flow using genetic markers, this is all microsatellite data, we've also done with SNP data, you actually find that there is massive gene flow across that gradient of that Drosophila species such that there's no genetic differentiation occurring. So the population structure in the species is very weak across that range, that southern border range. So we have a situation where we think this soft selection is actually kicking along and stopping the species from spreading further. So hard borders, soft borders driven by population processes, and hard borders sort of very firmly hitting this sort of genomic limit. So, you know, this is a paper that John Bryan and I put together quite recently that sort of pushes this sort of notion a bit harder. So we think about soft limits, we talk about hard limits, and from an environmental adaptation point of view, it's very, very important to recognize these limits. Because if you've got a hard limit, it doesn't matter what you do genomically, you're going to basically be stuffed from an evolutionary point of view. There may be no evolutionary adaptation under that hard limit. So we tend to focus on the left-hand side, but we should also focus on the right-hand side a lot more, I suspect. So like everyone, of course, we started with soft limits, and what we did is we did a huge amount of work on gradients, on climate gradients. And this has become very trendy these days because people want to actually use genomics to investigate genetic changes along gradients in all sorts of species. The advantage of Drosophila, of course, is that you can do detailed experiments alongside establish these gradients along these climate pathways. And that's exactly what we've done. So basically, we've characterized climate adaptation along gradients, and we've then dealt with it genetically, focusing on single genes, but then also very much doing genomics in the recent studies. So I don't have to talk about this very much because Carol explained this beautifully. Common garden experiment, very similar to what she had. So you've got a situation where you're really seeing things across a couple of generations, and, you know, and to be consistent with Timothy's talk, we're also looking at carryover effects, you know, epigenetic or other carryover effects. So that's why we go a couple of generations before we establish the genetic basis of these clines. And, you know, over the years, we've shown clines in a whole lot of species and a whole lot of traits. So here's an example of Drosophila melanogaster showing a cline in heat. So as you go to the tropics, you become more resistant to heat knockdown than in temperate areas. And under chill coma, you see exactly the same, the opposite pattern, but again, very powerful, very strong. So, you know, we can say, oh, well, you know, the species might actually be able to adapt to climate change because you have a nice gradient here, and that's worth studying. The other thing to note, of course, is that you don't always get a gradient where you might expect one. So virgin retention, which is a trait that we expect to be selected along a cline, we never see a shift. And again, that might be a hard limit as opposed to one of these soft limits going on. So that's, you know, that's the situation that sort of by 2000s, we characterized all these things, and we started looking at the genetics behind them, and then, of course, the genomics after that. And we thought, look, this is going to be easy, right? Because basically what we can do is we can actually pick a few candidate genes, show that those candidate genes are associated with these traits, and then we can say, hey, we've learned something. So on a few occasions, we were able to relate very nicely these clinal patterns to specific genes. So here's the one that actually is the one that really stood out for me. So this is that work that Marina Tolonis did in the lab, quite a long time ago now. And what she was looking at was she was looking at pigmentation in these flies, and the pigmentation shows a very clear cline, such as that as you go down south, you basically shift your pigmentation pattern. And she was able to show that that entire pigmentation pattern was almost entirely due to the expression of a single gene, the ebony gene in Drosophila. Once you accounted for that, you basically had almost all the variation accounted for. So it's sort of reminiscent in the story that Carol just told us before in relation to the, those particular transporters that she was talking about. So very convincing. Many other genes, we didn't see that. Many other genes, we looked and we just couldn't get successful. And so here's one of our frustrating stories. So we had a gene called FST, it's called FROST, and it was named because it was upregulated under certain conditions, and we thought, wow, this is a good gene to go for. And of course, being Drosophila, we could actually knock out this gene. And in the old days, we could obviously do this through one of these RNAi approaches. And of course, nowadays, we've also done it in CRISPR. And we've done the same sorts of knockouts, not in this gene, but in other genes. So you knock it out, and what happens is that, the gene's called FROST. Wow, it must have something to do with cold resistance, right? And you knock it down, and yeah, look, these flies become less tolerant of cold stress. And, it's quite a decent effect in actual case. You can see that those two lines, the red and the blue line, differ massively in terms of the amount of flies that recover when you experience a cold stress. So you knock the gene out, and you basically become much less resistant. So we then thought, oh, this is pretty good. And so we then found that there was a proline repeat gene in this gene, and that was the really prominent one. And this proline repeat gene, the biochemist told us, hey, look, this is going to be associated with cold resistance. All you've got to do is characterize that. You've got your fantastic candidate gene, and you can go and write up your fantastic paper. And we thought, oh, it's pretty good, nice to get a good publication out of this. And of course, we did that, so we had these repeats, and these repeats showed beautiful clines. The 6 repeats and the 8 repeats showed opposing clines, so you were switching these alleles beautifully in this particular gene. And of course, we thought, great, there's your cold cline, we've got a solution, we've got a single gene that accounts for a lot of control cline. Of course, instead of writing it up, we then thought, oh, we better just do the association study to make sure it's actually real. Did a giant association study, we got no association. It's one of those examples where, you have a spurious correlation, right? And you've got to be very careful of those things. Always watch out for those. So, part of the fun, the way you go. So, of course, we've done this in a range of organisms, including, believe it or not, trees. So we had a project that Rebecca Jordan ran in our lab for a number of years, Eucalyptus spicacarpa, which I'll come back to, hopefully, if I get time. And, this was all done genomically, and you get these beautiful patterns. Oh, sorry, just very quickly. So again, this tree is critically important ecologically, and the distribution is very wide, and you get lovely aridity gradients, lovely temperature gradients, and you can associate those with a bunch of different SNPs. So there's an annual temperature, for instance, a bunch of different SNPs, and you can say, well, that's pretty exciting. And of course, what that does give you is evidence of some sort of adaptation going on, right? There's some sort of adaptation going on. You don't know whether the SNP is associated with a particular trait, but at least you've got evidence for adaptation. I think it's one of the nice things about genomics. So we then thought, well, let's have a look, and we had a genome of a related species. You throw them on there, and it's one of these, it's one of these classic examples, as Carol pointed out, of a polygenic trait, right? The genes are everywhere along the chromosomes. Damn it all, Anyway, it's life. So we know that we get these nice clines. We know there's temperature adaptation that's happened in this tree species, and we know, of course, that we have a situation where it's polygenic and spread throughout the genome. Now, this slide I put in yesterday, and it's kind of interesting because Carol actually alluded to something similar. And so she alluded to some work that came out of Schrotter's lab, and Schrotter's lab was trying to evolve these different Drosophila lines at different temperatures, and Carol mentioned before, well, this temperature is going to involve a lot of different genes, probably right, and that's probably why you're getting these quite polygenic responses. And of course, what that means, as she said, is that you can get these differences among replicate lines. in Drosophila showing these patterns. Now, we're looking at a much simpler trait, and we actually had done this work a little bit before the Schlotter lab published that paper in 2016. And what we showed was something really interesting. So this is a much simpler trait. You're talking about desiccation resistance, right? So you evolve these replicate lines and you make them have exactly the same level of desiccation resistance from the same base population. You've got hundreds of individuals in each replicate population, so you know you've got not much genetic drift going on these lines, and you then map them. And when you map them, this is what you get. And if you look at that very closely, you can see that despite the same phenotypic endpoint, those dots are all over the place. Sometimes you get in one part of the chromosome, sometimes the other part of the chromosome, and there's no consistency. What Pip Griffin that did his work also showed, and we debated this because it was quite frustrating, is this is kind of complicated, but what we then did was to say, look, maybe you've got a whole lot of genes that can actually respond to selection, and they all get pushed the same way, but for whatever reason, some do and some don't. So let's take every single gene that shows any pattern of shifting in a particular direction and look at the similarity among the replicate lines to see what actually happens. And so what we did was we then said, look, let's look for convergent patterns regardless of whether we get significant peaks. So any SNP that shifted in the right direction, let's actually make sure that we map that. So convergence means that in one replicate line, one replicate selected line, you've had convergence with the other replicate lines. And the one on the left is the control line, which shows a bit of evidence of lab adaptation. The one on the right is the selected line. And if you look at that, you can see that when you pick one replicate line and look at all the genes that actually show a signature of selection, and then ask how many of those genes that showed at least a little bit of a shift in the SNP in the right direction in all the other lines, forget about whether it's significant or not, or mapping it, you actually do get quite a good signature. The bars on the right are much bigger than the bars on the left. So what does that mean? Well, it means that we have a polygenic model and we think all these alleles are ready to go, and some of them go and some of them don't. Right? They all get pushed in the same way, but some, for whatever reason, it could be interaction effects, could be epistatic, etc. They're being pushed along in some cases, but not in others. And I think this is, this is a good way of looking at these sorts of traits. This really changed the way that we look at these sort of things. So a bit of a lesson for us in this particular situation. Replicate lines, they don't show the same signature, but they might show the same tendency for genes to be pushed in the right direction. And some go, some don't go, for whatever reason we're not quite sure about. Okay, what we've now done is we've now switched to not so much looking at the soft selection, describing selection response, but asking the hard question, what about this particular situation where we get a hard selection limit? What happens? Is it the case that we get a whole lot of expansions and contraction of genes? Is it the case we get loss of function? What's going on, and can we get a signature of it? So here's our model system. This is published way back in 2003. So we have, if we select a whole bunch of species, we actually get a selection response for traits in some species and not in others. And Drosophila birchi is a rainforest species, very strict in its distribution, and it doesn't respond to selection. We have a whole lot of other species that are also sitting at a particular point that do not respond to selection. In this case, for desiccation resistance, we show the same for cold resistance. So we have a way of partitioning species, and we then thought, well, let's look at this in detail by taking a whole range of Drosophila species and let's pump them through and characterize the genomes of a whole lot of species and see what they have in common. Look at the ones that actually have a broad climate distribution, the ones that don't have a broad climate distribution. We know that the ones that have a narrow distribution tend to not be able to evolve. What's going on here? Can we get a signature of this? So this is looking at this hard selection limit in a different way. So on the right-hand side here, we have the climate. So when you see those gray bars are going all the way across, we've got a situation where the species can occupy a range of climates. And when there's only one bar filled out of 3, you know that it's a restricted climate specialist. There's the phylogeny. These are the ones that have all been sequenced. A lot of those were done by us. Some of them were done by other people. We put it all together and we get a pattern. So what do we find? Well, what we find is that when you look at the species that have been able to occupy a range of different climate conditions, you get a peak in the number of genes that are being that are changing, either being deleted or being duplicated towards recent times. So here's actually the picture from Melanogaster. So don't go back too many million years, but if you look at the left-hand side of that graph, you can see that there's a peak going up very, very sharply. So we know that the genome of these widespread species like Melanogaster is changing quite a lot, and it's changed quite a lot in recent times. Compared to these narrowly distributed species, we don't see that sort of peak. What we then did was to also look at variation within these species by resequencing a whole lot of these and asking the question, well, can we get signatures of what might be happening? And we're looking for two types of signatures here. So the first signature we're looking for is simply nucleotide diversity. So is it the case that when you get a lot of nucleotide diversity, you actually get species that have a broader climate distribution as opposed to ones that don't? And the answer on the left-hand side is yes, you do, because basically we've got situations where these niche position species on the right-hand side, these restricted species, show a lower levels of nucleotide diversity. So it's amazing you get that signature, but you do it at the species level. Importantly, at another level, and this is basically a DNA decay, loss of function point of view, you also find a good signature, but it goes the other way. So the species that are restricted actually have a greater signature of DNA decay in their genome generally. So, I was quite surprised to see this. And of course, this, you might expect a much stronger pattern if you're looking at candidate genes, but I think there might be something in this in terms of genomically picking up the species that are at these hard limits in this sort of climate point of view. So that's where we've got so far. So what I want to do now is to sort of charge through, the sheet hasn't worn up yet, that's good. So I just want to charge through a couple of applications because, of course, what we have to do is we have to try and do something now. There's no point plotting the end of the world in more detail. You need to do something now. So what can you actually do? And so what we've really started doing is to go back towards genomic variation, heterozygosity, as a measure that people can initially use, but then also looking at genomics where it can actually plug some interesting gaps. So the reason why we focus on genomic variation is obvious. There's a whole lot of data out there that some people have forgotten about, but hopefully most people remember that. If you have a lot of genomic variation, you're going to respond more quickly to selection. If you've got selection that you don't expect to happen, if you've got lots of variation, you're going to be able to do it more quickly. So this is an example of some detailed fly work that we collected with the Danes, fairly recently. It was published a couple of years ago. And it shows very clearly that when you've got lots of genomic diversity, lots of nucleotide diversity, you're going to respond quickly to selection. So that's great, fantastic signature, beautiful signature, you get it very tight. So what this does is it asks the question, well, what should we be looking for in species, and how should we be treating them, particularly in terms of mixing things up, in terms of where we go into the future, right? How do we preserve biodiversity from an evolutionary point of view? How do we make sure that species have the potential to adapt? And, people over the years have come up with different indices. The classic one is, of course, overall genetic variation. Make sure they've got lots of genetic variation. We can measure it better now with genomics. Hence, you're going to be able to adapt, and you're going to be able to do quite well in the future. And, but there's other ones that are coming on board that are interesting, but they haven't yet been proven. So one of them is Future Adaptation Index. So that's obviously one where you look at the candidate genes and the allelic shifts in the candidate genes to be able to make some predictions. You're going to need a good set of candidate genes, of course, to be able to do that. And the question is, how good a candidate gene set do you need to be able to do that? And of course, the other thing that people are saying to do is to look at population adaptation index. So how unusual is a population in terms of outlier genes? If you can identify a population that has a lot of outliers, do you think that's the one that we should focus our attention on because it might have a better adaptive potential in these particular situations too? Okay. So we've done that, and we've worked on several marsupial species, and in this particular case, this is quite a famous case now, where we showed that when we took a low genomic variability population and we introduced material in it that shoved it up, and this was a threatened population, it's a threatened species, very threatened species, we were able to boost its population size massively by doing that particular effort. And this is a species that in the future, in the next few years, faces the loss of snow, and that's pretty important. And what we're finding is the heterozygosity is obviously shooting up, and now it's leveled off, and it's leveled off very nicely at a level that you're much happier about. The other situation we've dealt with is where people have come up to us and said, look, everything's dying. What are you going to do about it, right? So the macrocarpa, that Eucalyptus macrocarpa I mentioned here, has situations where some populations are actually dying. And this is a revegetation site that was put out by a whole lot of volunteers, and the whole area just died off. And this is due to droughts and high temperatures combined. So what we've then said is, well, we need to get creative about this. We need to bring some genotypes in from the outside to make the future. So you've got a situation where you've got this area that we are introduced. This is the area where all the trees died. We then introduce genetic material from very, very broadly and bring it in. And we know, based on genomic data, that this is pre-adapted to dealing with this. Yep. So to cut it very short, did this complex design, bring a whole lot of provenances. So we're trying to secure these trees for the future. And we then have an excellent scientific experiment as well, where we can actually measure their growth the next time the major drought hits. And that's what we, of course, intend to do. So these are the things that I think are very important for setting up. So that's my range of points. Hopefully, I've at least convinced you of some of those. I think we need to act now. We can't wait too long, even though we'd like to. And the future is grim, but the future can be tackled, at least if we are willing to take a few risks. So that's where I'll leave it.